So, so the summary of the sexual reproduction in flowering plants, that is the angiosperms. So the summary: the flowers are the seat of sexual reproduction in angiosperms. So in the flower, androecium consists of stamens representing the male reproductive organs, and gynoecium consisting of pistils. So gynoecium consists of pistils representing the female reproductive organs. Okay. A typical anther is bilobed, it is diathecous, and it is tetrasporangiate. Now pollen grains develop inside the microsporangium. Now pollen grains develop inside the microsporangia. Four wall layers, the epidermis, endothecium, and middle layers and the tapetum surround the microsporangium. Four wall layers, the epidermis, endothecium, middle layer, and the tapetum surrounding the microsporangium. Right. Now cells of the sporogenous tissue laying in the center of the microsporangium. So the cells of the sporogenous tissue laying in the center of the microsporangium, right, undergo meiosis, that is microsporogenesis, to form tetrads of the microspores, right. Individual microspores mature into pollen grains. So when the individual microspore mature into pollen grains. Pollen grains represent the male gametophytic generation. Pollen grain represents male gametophytic. Pollen grains represents the male gametophytic generation, right. The pollen grains have two-layered wall. The outer is exine and inner is intine. The exine is made up of sporopollenin and has germ pores. So the exine is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and a generative cell, or three cells, a vegetative cells and two male gametes at the time of shading. So pollen grains may have two cells, that is a vegetative cells and generative cells, or three cells, a vegetative cells and two male gametes at the time of shading, or three cells, a vegetative cell and two male gametes, a vegetative cells and two male gametes at the time of shading. So shading means it's like the so when the pollen grains are shed, it means it is, so when the pollen grains are shed, it means it is present on the stigma. Okay. So the pistil has three parts, that is the stigma, style, and the ovary. So the pistil has three parts, the stigma, style, and the ovary. So ovules are present in the ovary, right. The ovules have a stalk called funicle. So ovules have a stalk, it means ovules have a stalk called funicle, protective integuments, and opening called the micropulse. So it has a stalk called funicle, and then protective integuments, and an opening called micropyle. The central tissue is the central tissue is the nucleus, in which the archaeosporium differentiate. So the central tissue is the nucleus. So the central tissue is the nucleus. So the central tissue is the nucleus, in which the archaeosporium differentiates. A cell of the archaeosporium, the megaspore mother cell, divides meiotically. A cell of the archaeosporium, the archaeosporium differentiates. The central tissue in the nucleus, in which the archaeosporium differentiates. A cell of the archaeosporium, the megaspore mother cell, divides meiotically, and one of the megaspores forms the one of the megaspores, it forms the embryo sacs. Now the female gametophyte and male gametophyte, that is the pollen grain. Okay, the mature embryo sac is 7 cell and 8 nucleate. So mature embryo sac is 7 cell and 8 nucleate, right. 7 cell and 8 nucleate. So at the micropylar end is the egg apparatus, consisting of two synergies and an egg cell. So at the micropylar end is the egg apparatus, consisting of two synergies and an egg cell. Okay. So the egg apparatus consists of two synergies and an egg cells. So at the chalazal end there are three antipodals, and at the center is a large central cell with two polar nuclei. So at the center is a large central cell with two polar nuclei, right. Now the pollination is the mechanism to transfer pollen grains from the anther to the stigma. So pollination is the mechanism to transfer pollen grains from the anther to the stigma. So pollinating agents are either abiotic, that is wind and water, and biotic, that is animals. So pollination agents could be abiotic, that is wind and water, and biotic, that is the animals, right. So pollen-pistil interaction involves all events from the landing of the pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. So the pollen-pistil interaction involves all the events from the landing of the pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. So there can be the rejections, right, between the pollen and pistil. Following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style. So following compatible pollination, the pollen grain germinates on the stigma. So following compatible pollination, the pollen grain germinates on the stigma and the resulting pollen tube grow through the style. So following compatible pollination, pollen grain germinates on the stigma and the resulting pollen tube grow through the style, right? Enter the ovules and finally discharges two male gametes in one of the synergies. Right? So angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and the triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. So, uh, so okay. So the products of these fusions are the diploid zygote. So the products of these fusion. So the products. So the products of these fusions are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. So the products of these fusions are the diploid zygote and the triploid primary endosperm nucleus. So in the primary endosperm cell, right? Zygote develops into. So the zygote develops into the embryo and the primary endosperm cells form the endosperm tissue, right? So the primary endosperm cells, as we can see, the triploid primary endosperm nucleus, right? So in the primary endosperm cells, so zygote develops into the embryo and the primary endosperm cells forms the endosperm tissue, right? The formation of endosperm always precedes development of the embryo. So formation of the endosperm always precedes, so it precedes first development of, always precedes the development of the embryo. So the developing embryo passes through different stages such as the proembryo, globular and heart-shaped stages before maturation. So the developing embryo passes through different stages such as the proembryo, globular and heart-shaped stages before maturation. Okay. So developing embryo passes through different stages such as proembryo, globular and heart-shaped stages before maturation. So mature dicot embryo has two dicotyledons and embryonal axis with epicotyl and hypocotyl, right? So with two cotyledon and an embryonal axis with epicotyl and hypocotyl. So epicotyl is at the tip and okay. So where it is like, so epicotyl and hypocotyl, right? So embryos of monocotyl. Okay, epicotyl and hypocotyl, the embryos of the monocot have a single cotyledon, right? After fertilization, ovary develops into fruit and ovules develop into seeds. So after fertilization, the ovary develops into fruits and the ovules develop into the seeds, right? A phenomena called apomixis is found in some angiosperms. Particularly in grasses. It results in the formation of seeds without fertilization. Because apomixis, it results in the formation of seeds without fertilization. Now, apomictics have several advantages in horticulture and agriculture, right? Now, apomictics have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seeds. This phenomenon is called the polyembryony. So, more than one embryo in their seed, the phenomena is called the polyembryony, right? So we are going to study the exercises. So name the parts of an angiosperm flower in which development of male and female gametophyte takes place. So name the parts of the angiosperm flower in which the development of male and female gamete takes place. So male gamete in the pollen grains and a female gamete in the female gamete in the in where, like female gamete in the you can see pollen grain, and female gamete in the ovule, right? Now differentiate between microsporogenesis and megasporogenesis. So microsporogenesis is a formation of a tetrad, macrospore tetrad, and megasporogenesis, that is formation of only one, formation of only one, you can see one egg or something like that. Which type of cell division occurs during these events? Name the structures formed at the end of these two events. So which type of cell division occurs during these events? So during these events there is um, to differentiate it, at first it is like the for the differentiation it is like the first mitosis, and then after there is a meiotic division, that is meiosis, right? That is meiosis. So name the structures formed at the end of these two events, right? So which type of cell division occurs during this event? Name the structures formed at the end of these two events. So name the structure, like name the structure, what? Male gamete and female gamete. What it is like? Arrange the following terms in the correct developmental sequence. So pollen grain, microsporogenesis. So arrange the following terms in the correct developmental sequence. So pollen grain, sporogenesis tissue, microspore tetrad, pollen mother cells, and male gamete. So arrange the developmental sequence. So first there is the sporogenous tissue, then it has a pollen mother cell, then they form the so sporogenous tissue, then pollen mother cells, then the pollen mother cells form the microspore tetrad, and then there is like the pollen grain, and then there is the male gamete, right, by the generative cells. So with a neat label diagram describe the parts of the typical angiosperm ovule. So you know the ovule like stigma style, and ovary. So ovule, it has the integuments, and then there is like it has integuments, and then there is like then there is like what, there is like nucellus, and inside that there is like embryo, and then there is like embryo sacs, right? Then there is like egg apparatus, and on the opposite side there are antipodal cells, and all that at the chalazion, and micropole, and has all these. So this is it, like a typical angiosperm ovule. What is meant by monosporic development of female gametophyte? So what is meant by monosporic development of female gametophyte? It is because it is developed from only single megaspore, right? So this is the monosporic development of female gametophyte, right? Now with a neat diagram explain the 7-celled and 8-nucleate nature of the female gametophyte. So 7-celled and 8-nucleate nature of the female gametophyte, right? What are the chesmogamous flowers? Can cross pollination occur in cilistogamous flowers? Give reason for your answer. What are chesmogamous flowers? Can cross pollination occur in cilistogamous flowers? Chesmogamous flowers, so they are the open flowers, and cilistogamous flowers, they are like the closed flowers, right? Give reasons for your answers. Can cross pollination occurs in cilistogamous flowers? No, because they are open. No, because they are closed. And what are chesmogamous? Yeah, they do have the cross pollination, right? Mention 2 strategies evolved to prevent cell pollination in flowers. So it is like the pollen incompatibility, right? And then if they have the different synchrony between the development of the pollen grain and the ovary, that is receptive, that is stigma, right? So this is the cell incompatibility. Why does cell pollination not lead to seed formation in cell? Okay, so this is the cell pollination in flowers. If the flowers, they are to prevent, if the flowers should be like unisexual, it has only stamen, or either have the carpels. So what is cell incompatibility? It is the rejection. The pollen is not compatible with the stigma, right? Why does cell pollination not lead to seed formation in cell incompatible species? Because the pollen just not going to germinate until and unless if it's compatible, and it's compatible it should be of the one species, the similar kind of species. Okay, now what is bagging technique, how is it useful in a plant breeding program? So what is bagging technique? So bagging it is like, so bagging, so when so after the bagging there is like for the pollination, so the unwanted pollen should not be pollinate that thing. Okay, so how is it useful in a plant breeding program? What is triple fusion? Where and how does it take place? Name the nuclei involved in a triple fusion, right? So what is triple fusion? It involves like the three nuclei to get fused, like two that are the central nuclei. It has two polar nuclei, and then there is an one male gamete, so this is the triple fusion. Where and takes place in the embryo sac, and how does it takes place? You know it very well. Name the nuclei involved in triple fusion. Two polar nuclei and one male gamete. Why do you think the zygote is dormant for some time in a fertilized ovule? Why do you think the zygote is dormant for some time in a fertilized ovule? Differentiate. Differentiate between hypocotyl and epicotyl. Say epicotyl above the soil and hypocotyl below the soil. Coleoptile and coleorhiza. Coleorhiza that covers the roots, and coleoptile that covers the plumule. Integument and testa. Integument it for the protection. Testa is it is and also the testa and tegmen. So testa is also the, I think it is also the ball of the seed, right? Testa inner is the tegmen, testa is the outside, right? The perisperm and pericarp. So perisperm it is the remnants of nucleus that acts as a endosperm, and pericarp it is the it is a fruit wall, right? Pericarp it is a fruit wall. So why is apple called a false fruit? Which parts of the flower forms the fruit? So which parts of the flower other than ovary, or if the fruit is the if the fruit is the apple, then it is also from the thalamus, right? And why is apple called, so this is why it is called the false fruit, because true fruits only form from the ovary. Okay, so what is meant by the emasculation? So it is the removal of the anther tip so that the pollination does not happen from the same species, or self pollination kind of thing. So when and why does a plant breeder employ this technique? Okay, to prevent the self pollination, right? To increase the variation we need the cross pollination. So this is why. If one can induce parthenocarpy through the application of growth substances, which fruit would you select to induce parthenocarpy and why? So fruit there are like tomatoes, you know the parthenocarpy in tomatoes, right? By the auxins. Then yeah, to induce, and then so this is it, like parthenocarpy, induce parthenocarpy in tomatoes through the application of growth substance. Which fruits would you select to induce the parthenocarpy? Or banana, I think parthenocarpy fruit is well known is a banana, right? It is a seedless fruit. Explain the role of tapetum in the formation of pollen grain walls. So explain the role. It nourishes the developing pollen grains, right? And it is multinucleated, and that's it, like. So okay, and and it is the innermost layer, that tapetum is the innermost layer. So what is apomixis? It is a formation of seeds without the fertilization. And what is its importance? I mean, you don't have to buy the seeds and all that, like the hybrid seeds and all that things. So this is your chapter is done in here, that is sexual reproduction in flowering plants.